Okay, now let's look at the process of import and export at a molecular level in more detail in order to understand how this process is working. So we have talked about this. The nuclear import receptor binds the protein which has NLS. This binding causes a conformational change. This receptor then binds the nucleoporin. These are the proteins that make up the nuclear pore. And we have said that lining, this lining is basically is made up of FG repeats, phenylalanine and glycine repeats. And phenylalanine and glycine are repeated all along this red zone that I'm drawing. So this basically receptor attaches to FG repeats, dissociates. For example, it is bound to FG repeats here. It will dissociate here and it will bound the FG repeats here, and then it will dissociate from FG repeats here too and bind FG repeats at this level. And it will, this process of association and dissociation with SG repeats will continue, and ultimately this basically receptor will enter the nucleus where we have our RAN, where we have our Jeff, which has produced RAN GTP. When RAN GTP binds this receptor, it will cause unloading of our cargo, and this cargo will be now a resident of nucleus. So this basically is how the nuclear import receptor is able to bring the protein which has NLS into the nucleus. And since we have RAN GTP in nucleus only, we talked about that in previous module. Since we don't have RAN GTP in the cytoplasm, this gives basically directionality to this process from cytoplasm direction into the nucleus. Otherwise, movement of this receptor dissociation is a reversible process. It can go in both directions. It does not go in both directions. It is favored in the direction towards inside the nucleus because of presence of RAN-GEF, which is exclusively present in the nucleus and not in the cytoplasm. Once the cargo has been unloaded, RAN-GTP, when it binds the import receptor, it again causes a conformational change in this receptor, and now it can bind the FG repeats. Remember, our import receptor cannot bind FG repeats unless it is attached to the cargo protein. Here we don't have the cargo protein. RAN-GEF is sort of supplementing the function of cargo protein, allowing this receptor, import receptor, to bind FG repeats. Again, this receptor crawls its way through and goes to the cytoplasm. We have RAN gap, which will allow the RAN to hydrolyze GTP into GDP. This receptor cannot bind RAN GDP, and it will become unloaded, and now it is recycled and it is ready for the next protein molecule which has NLS. So this is how basically the nuclear import is functioning. So we have talked about all this process in detail. Now let's look at the nuclear export. There are proteins that have to be exported outside the nucleus. From the direction is from nucleus into the cytoplasm. The nuclear export receptors work the same way but in reverse. So nuclear import receptors can bind their cargo only and only if they are attached to RAN GTP. So RAN GTP will bind the nuclear export receptor, which will go through conformational change. Now it can bind, our export receptor can bind a protein which has export signal on it. This complex will basically again now crawl into the nuclear pore and bind FG repeats, and it will crawl. This whole complex of 3 molecules will go into the cytoplasm, where we know that RAN gap is present. RAN gap will make RAN to hydrolyze GTP into RAN GDP. Since this export receptor cannot bind RAN GDP, it only binds, it has preference for binding RAN GTP. RAN GDP will dissociate, and the condition for this receptor to bind the cargo protein is that it has to be bound to RAN GTP. Now we have taken the RAN GTP off this receptor. The cargo will be unloaded and it will be delivered to the cytoplasm. This receptor, again without the RAN and without the cargo, it does not like living in the cytoplasm. It will now bind the FG repeats and it will crawl back its way into the nucleus, and the whole process will start again once again when this receptor finds RAN GTP and protein which has nuclear export sequence on it. So this is both the processes in comparison, and so you can see we have talked about this. Basically, driving factor here is the presence of RAN assisting molecules. RAN gap is present in the cytoplasm, and RAN GEF is present inside the nucleus, which is basically giving directionality to this whole process. So we are next going to talk about how once the cell divides, the nuclear membrane dissociates, and how is cell able to selectively import molecules into the nucleus when that happens at the cell division. We'll talk about that next.